Well, I think we have to switch gear a little bit after this exciting talk from Nancy, where she looked at basically the whole spectrum of markers for chronic fatigue. I am looking at one specific retrovirus and the expression profile of one specific retrovirus. So that's reflected in that I'm a basic scientist. I've worked for the last 10 years on this particular retrovirus, which I have discovered, which encodes the superantigen, and I'm going to explain you all this in great detail. And it has come as a second way for me to actually get involved in chronic fatigue. So I'm going to speak about endogenous retroviruses. That's my main work. This human endogenous retrovirus, HERV K18. That's what I've done for, as I said, over the last at least 10 years, but we've only fairly recently actually used this information which we've gained about this virus to apply to chronic fatigue patients. And of course, we also look at XMRV, the infectious retrovirus. Of course, we only do this since Judy's paper came out in October, which was very exciting for all of us. And I'm going to tell you where we are in our analysis of this. So basically what I wanted to point out to you is that I'm speaking about two completely different retroviruses that have the same function, but one of them is in our genome, is inherited as a Mendelian gene, and the second one is an infectious virus, as you know from Judy's work, which comes from the outside world. So what I want to see is if any of these retroviruses either cause, trigger, or sustain chronic fatigue syndrome. So let me give you a little bit background on human endogenous retroviruses, or HERVs. That's the abbreviation which is known. Now, these retroviruses overall are footprints of previous exposure to retroviruses. So basically, over evolution, infections have happened over and over, and in very, very rare cases, an infectious virus actually hits a germline cell, either a spermatocyte or an oocyte, and at that point, this retrovirus will now be in the germline. It will be inherited as an Mendelian gene. It has a very, these human endogenous retroviruses have very similar organization as other, like HIV, any other retroviruses, 5' 3' LTR and GAG, POL, and ENF. However, most of them have accumulated many, many mutations, and none of them are known to make complete viral particles. Nevertheless, it's known that the LTR regions from these endogenous viruses still have promoter activity, and depending on where they integrate in the genome, they can actually turn on higher level of expression of various genes, and we have many, many examples of that. And the main point which I want to just stress again, so these HERVs are transmitted vertically through the germline. So we all have these HERVs. But what I have found is that the level of expression is where this retrovirus is controlled, and that's what I'm going to tell you. First of all, I want you to know that almost 10% of the human genome is actually retroviral elements, which is a shockingly high number, considering that we know absolutely nothing about any of them and what they do. We know about some promoter elements that turn on genes in various diseases. But there's clearly here is a gold mine which has basically been untouched. And it has been untouched because it's so difficult to actually identify particular retroviruses because there's so many integration sites. I've already mentioned HERVs are unable to produce active viruses because they have many, many mutations. But still, you can find transcription and translation of some HERV genes within the genes. So what I mean is they can't assemble a complete infectious virus, but they can make various viral proteins. And for example, what you could envision is if you get infected with a retrovirus, that you can have recombinance happening between an endogenous retrovirus and an infectious virus. And the family of the HERVs, which I'm speaking about, is HERV-K. It's the youngest family of HERVs. It has been integrated. We know exactly when it got integrated. It is when the old world monkeys and new world monkeys were separated because we can find this K18 in primates in Africa, but not in New World monkeys, just as marmosets. We know the complete position of this retrovirus. It's on chromosome 1, and it actually sits in a gene, CD48, which is a gene immunologists have been interested for a long time. CD48 actually was characterized and initially described as the first gene which is induced after EBV infection in B cells. It has the reverse orientation to the CD48, so this gene sits in the first intron of CD48, and we have never known that. That intron just has never been mapped. So basically what you have to envision that many, many other genes have retroviruses in it which haven't been identified. But the CD48 I'm going to come back to a bit more because that's the relevance with EBV, the EBV connection. As I said, the orthologs of K18 are found in old world monkeys. So we know exactly when this integration happened because it's not in the New World monkeys. We all have this retrovirus. The main characteristics of this retrovirus is that it's very, very low expression level. So basically, in a healthy individual, we can see only very low baseline level expression. However, we found that this retrovirus can be induced to be transcribed very easily. And the first one which we found to do this is this Epstein-Barr virus. And that was actually our connection with CD48, because as I said, CD48 itself is induced by EBV. More recently, we found also another herpes virus, HHV6, can lead to the transcriptional induction of this retrovirus. And with our collaborators, we have shown that interferon alpha induces the expression. And as you know, interferon alpha is a factor which is induced by many, many infectious agents. So you can see many different ways that this gene could actually be induced. We think it's kept, and the main reason why I'm really interested in this gene is because the envelope gene of this retrovirus encodes what we immunologists call superantigen. Superantigen, I'm just going to explain you in the next few slides. It's a very, very reactive molecule which activates basically the whole population of T cells. So it basically was these two connections, EBV. EBV has been linked to chronic fatigue since the beginning. Nobody can really find a complete hard correlation with it, and we felt that would make perfect sense if it would act through K18 expression, because so many other things could induce this K18 expression. So just bear with me, but I'm showing you in a cartoon fashion what the superantigen is. As you probably know, conventional antigen is presented by MHC molecules on a cell called antigen-presenting cell. And this conventional antigen is broken down in a very small peptide, and it's recognized by the T cell through both V regions of the alpha chain and beta chain with very, very high specificity. So in other words, if you look at the precursor frequency to a conventional antigen, it's probably 1 in 10,000 T cells. And so that's why you never see a primary immune response to a conventional antigen. Now, superantigens, which were defined at the beginning of the 1990s, are unprocessed molecules, and they basically completely circumvent this high specificity. They interact with all MHC molecules from the outside, and they interact with the T cell receptor also from the outside, therefore giving a massive activation. And it leads to so much activation that you have lots of bystander activation as well. In addition, both antigen-presenting cell produces cytokines when this interaction occurs, and of course the T cell makes tons of cytokines. And this really was the hallmark which we got very excited about, and we felt it fit very well of what we had heard, like from Nancy and from many other people, about this outpouring of many, many cytokines in these chronic fatigue patients. It could be that maybe there's abnormal expression of this endogenous retrovirus, and therefore the immune system is going haywire. So just a little bit quickly about the hallmarks of superantigens. So far, the only products are from microbial origins, either bacterial products, and the best known are toxic shock syndrome, or for food poisoning, SEA. These are bacterial proteins which give very massive T cell activation, and then there are these viral genes. And the prototype of this is the mouse memory tumor virus. That's where my work started in this field. And we have, from that work, we actually looked for human homologs, and then we came to this K18. The main point, again, I want to stress is that these superantigens have this rapid and massive activation of naive T cells. And that's really what you need to remember. It has T cell V beta specificity, it's class 2 dependent, there's no antigen processing. So here is basically our model system. We have the B cell, which has this K18 in its genome, CD48 in its genome. But this gene is silent, it's not turned on, till it gets a signal from either EBV binding to its receptor, or EBV infecting the cell. It's actually various mechanisms which we have identified separately, which turn on K18 specifically. More recently, as I said, it's also HHV6, another herpes virus, which has the same function, as well as exogenous interferon alpha leads to the expression of this superantigen. So we get now on this B cell suddenly a high amount of this superantigen expressed, which leads to massive T helper cell proliferation. And what we do know for EBV actually requires the superantigen to get to a memory B cell state where we have persistent infection and EBV can hide out and is no longer expressed. So it needs this T cell help. An EBV-infected B cell needs this T cell help to actually get to this point. So it's really required for the EBV reservoir, and we actually have anecdotal evidence for that. If you look at marmosets, New World monkeys, which do not have K18, however, the B cells are very similar to human B cells. They can be infected with EBV. However, what happens in these marmosets is that there's no persistent infection. We all harbor EBV. We all have EBV in us in a latent fashion, but marmosets cannot get to that stage. They can only get to the stage of lymphoblasts, where cytotoxic cells are very, very active and can actually prevent the outgrowth of these cells. And so we know in humans very well, if you have immunosuppression, you take away the cytotoxic cells, normal, healthy people can develop EBV lymphomas, which is seen very frequently in transplant patients. And so what we predict then also is that there may be some mechanism in chronic fatigue patients that these T helper cells stay activated because maybe these T regulatory cells are missing something so that we get this abnormal immune activation. I also want to point out that we know when we look at the human population that this K18 has some polymorphism. We can identify 3 alleles, K18.1, 2, and 3. So have single nucleotide polymorphisms. I'm showing you this because there's a peculiarity in that one allele, K18.3, is expressed relatively rarely in the Caucasian population. It's around 10%, whereas the other 2 alleles are found equally. And allele 3, for example, is not found at all in the Chinese population and is much lower in Asian countries. So our first hypothesis was that maybe this K18.3 had some special superantigen activity, and maybe we should type patients for the alleles and would see a correlation. And so basically then the working hypothesis is that K18 could be a risk factor for chronic fatigue. Turning on, I should say, of K18 is a risk factor for chronic fatigue. So the first, as I said, is we thought there could be differential allele distribution, that maybe allele 3 had some detrimental effects. And we have done this study, completed this study and actually published it in MS patients, where we show that huge cohorts of MS patients, over 1,000 patients compared to almost 1,000 controls, we can see that this allele 3, either 1.3, 2.3, or 3.3, have a higher risk to obtaining multiple sclerosis compared to the individuals who have other alleles. So that was very, very exciting to us. And we had actually started to do this also in chronic fatigue patients with mono, and we've seen initially similar tendencies. What stopped us actually going on with this work is this absolutely high numbers which you need to get functional significance. These are population studies. To see actually a penetrant phenotype, you need to get into thousands, and we realized that we could never get to this level in chronic fatigue patients. So then we switched gears, and we thought we look at differential expression of HERV-K18. And basically, as I already said, healthy patients have very low level of this retrovirus. And what we had noticed originally a few years back when we had started some chronic fatigue studies, that chronic fatigue patients in general had higher level of HERV-K18 expression. And we felt that was interesting, and we should really follow this up very systematically, not just like taking random patients. And so we decided to actually compare HERV-K18 expression in chronic fatigue patients and healthy controls, but with specific emphasis of chronic fatigue patients which are somehow linked to EBV. And we actually are funded, believe it or not, from NIH in a 5-year grant, and Renee Taylor is my co-investigator in this study. So Renee Taylor is a psychologist in Chicago who deals with patient selection and patient selection. So we have started in the last 3 years to assemble new chronic fatigue cohorts. It's a very, very slow process. I can only give you an update where we are. We have a group which we selected, which developed chronic fatigue after they had infectious mononucleosis. That is, after they had been exposed to EBV as adolescents, came down with infectious mononucleosis and could not resolve infectious mononucleosis. And Renee Taylor had studied such patients already for a while and have found a high significance with chronic fatigue patients. So we felt this would be the ideal group for us because we had the EBV link here. And then compared to this, we have chronic fatigue patients which are not linked to EBV. And then we have a very ambitious goal of following these patients every 6 months, draw blood from them every 6 months, so we could actually do these longitudinal studies, which you have heard so elegantly from Nancy. We are really only just at the beginning, but I'm going to show you what we have, and we already have some direct insights into this. So basically, what we would like to answer is, does the level of expression of HERV-K18 correlate with disease severity? We don't have the full answer, but we have clear hints on this. So here I'm just telling you the cohort which we have selected. This is basically all Renee Taylor's work. She interviewed 128 patients. Some of them have withdrawn. So in the end, we ended up with 105 subjects who were willing to, over a 5-year period, to coordinate with us to actually go through these various processes of donating blood. All of these 105 patients have been verified as having chronic fatigue by at least one physician, but we actually, each patient will go through 3 physicians' evaluation because we realize very clearly that our data will only be as good as the clinician's evaluation is. We have also baseline data and medical records from these 105 patients. So we have, from these 105 patients, 53 subjects have developed mono after EBV mono. 45 subjects have chronic fatigue through other onsets, unknown onsets, but never had mono. And 7 we haven't really fully identified yet. So the 6 months follow-up of this started last year, in August of last year, and basically from the 105 subjects enrolled, 49 have undergone the 6 months follow-up so far. We have done a 12 months follow-up. We started this February, and of course it's going to go on. It's going to in September again and next year. So we will have to wait till we have the complete outcome of these studies. So we have 25 subjects which have 12 months follow-up. So we have actually 3 blood samples from these patients. And as I said, we also plan 18 months and 24 months follow-up if the patients cooperate. Now, the technique we use is a highly quantitative method of qPCR, Tachman qPCR, where we measure the level of expression in relation to the 18S gene, which is done in the Tachman reaction, is done in the same tube. So there can't be any variations in sample size because the reference point is from the same RNA. And so basically, we establish a ratio of this 18S ribosomal RNA compared to K18 RNA, and that's how we express the level of expression. So the numbers of samples which we have tested so far, we have done 7 patients which had mono. From these 7 patients, we have 3 bleeds, so that's 21 bleeds. I know this is very, very low numbers, but when you look at the data, it's actually surprising what we could already see from this. We have 10 patients which did not have mono, and from 6 of these, we have a second row, but not a third row yet. And then a healthy controls, we have 14 just random people. So, as I said, we do this qPCR and we get to a ratio. And what we do is we use 1 reference sample in every reaction, which is always the same. So one healthy individual, we have made a lot of RNA. We use the same RNA in each assay as a reference point, and we have arbitrarily set the ratio for that person as 1. The ratio of 18S RNA to K18 RNA as 1. And so what you see is increases in ratio between in the different groups. So we have 14 samples, healthy controls, and overall the ratio is about 2 in this small group of patients. Then we have 9 samples of chronic fatigue patients. These are patients which did not have any signs of mono. We clearly see an increase. It's just barely significant, and obviously we have to do many, many more samples to do this. And then we have 7 samples. That is the first bleed from our chronic fatigue cohort. And you can see that of the mono patients, you can see there's clearly a much, much higher level of expression here. And that's really very, very impressive. An increase of this retroviral gene is functionally very, very significant and could really have strong functional consequences. So as I said here in this slide, we compare the first bleed of each one of the groups. And what we do know is that all the chronic fatigue patients in this group and in this group definitely at that time point had active chronic fatigue. They had the symptoms of chronic fatigue, they were interviewed, and that's how they were selected. So we know that at this time point, this level of expression correlates with disease. What was very shocking to us is when we started to look at the second bleed and the third bleed. So here now I'm showing you only the data from the 7 patients which had chronic fatigue after mono. So this, again, is bleed number 1, where we see very high-level expression. We measured 6 months later, so at the 12-month point, and we see complete normal expression in these patients. We really couldn't believe this data initially. We have done it many, many times, completely controlled. We reproducibly see this change in expression. And then 6 months later, it seems like to go up again. And of course, I mean, this is a very crude way to look. What we have to do is now go to every individual and look at the level of expression and at the particular symptoms at that particular time, which, of course, we haven't been able to do yet. And, but it's most patients at 12 months after initial chronic fatigue go through a period where they feel better. So we feel like this is a very strong functional correlation. This is the first and second bleed from the conventional chronic fatigue patients. We see also a reduction after 12 months. Again, here at this stage, at the first bleed, this was active. This patient had active disease. We don't know each one. They were all interviewed, but basically we haven't opened our, everything is blinded and we don't really know yet. We have to go in more detail. So the conclusions we draw from this very, very little data which we have so far, that K18 mRNA expression is upregulated, both just in normal chronic fatigue patients compared to healthy controls, but even much more significantly in EBV IM chronic fatigue patients compared to healthy controls. And secondly, equally important is that the K18 mRNA fluctuates over time in these patients. So I want to put this out as a warning. It's really, really essential that when anybody is doing any studies on blood cells from these patients, that at the same time there's a clinical evaluation of the patient, because an individual patient can go through a lot of fluctuations, and this is kind of extreme what we've seen. And we're actually actively pursuing this to see if there's a direct correlation with disease symptoms. We're starting a cohort similar to what Nancy has described to you, with good days and bad days. We have a new study with Tony Komarov, who has a huge cohort of chronic fatigue patients. We're asking these patients to donate blood when they feel especially bad and when they feel especially good. So in other words, instead of just all 6 months, which is an arbitrary time point, we're going to take it directly at the time when disease is either high or low, and that will really directly answer this question. Now, just a couple of minutes, I want to tell you what we have done since Judy's paper has come out on XMRV in chronic fatigue patients. We were very, very excited when this paper came out, and we already had huge cohorts of samples, blood samples in our freezer, and so we instantly, of course, wanted to test our samples for the presence of XMRV. Now, what we felt what would be the best assay to do is to use a highly, highly specific Tacman qPCR, which have been developed for the XMRV integrase gene. This assay has been developed by Ila Singh for prostate cancer, and it's been specifically developed that it does not cross-react with any mouse endogenous viruses at all, and that's a really, really important point. However, this assay detects any XMRV which has been cloned so far. So what I'm going to show you is that we have actually from WPI a cell line which expresses XMRV. We use that for our titration of genomic DNA. And this is our qPCR. In red, you see the 18S RNA. In black, you see the titration of DNA, starting at 1 nanogram down to 1 picogram. And basically what I'm showing you here is that we are able to see 10 nanogram of this cell line, 10 nanogram DNA from this cell line, which is the equivalent of a single cell. So the assay is very sensitive. We can show that we can detect from the cell line a single cell. We wanted to see, could we see the single cell if it was in a bed of negative cells? So we repeated the same analysis. Again, we used this WPI cell line, but now we added 5 microgram, which is 500,000 times more, control DNA, negative DNA, which we had before defined as not having any of this virus. And we could show, again, equally now in this assay, that basically we could see 10 picogram of this WPI DNA in 5 micrograms, so that's 1 in 500,000 cells. So the assay is exquisitely sensitive. Now, so this seemed to be the assay of choice because we wouldn't have to worry about any contamination, and it is obviously very, very sensitive. So we used 3 independent chronic fatigue cohorts because that's what we had collected already. These are not new studies. These are studies which had already been banked in our freezer. We have totally 228 samples. The first cohort we had is from Sue Levine from New York. She has a chronic fatigue clinic, and her patients are all characterized as having typical chronic fatigue with strong disabling function, but there is no infectious origin of, in these patients, and that's actually how we had studied the previous work on. Then, as I introduced to you in the previous slide, we now are starting this new cohort with Taylor cohort of 105 samples of chronic fatigue patients which got specifically a disease after infectious mononucleosis. And then we actually had also 11 samples from the HHV6 Foundation, and these actually were particular samples which had chromosomally integrated HHV6, and we wanted to see if there was a correlation on this, because, and in all of these samples, we could not see any XMRV integrase. So I know this is very shocking, and I know negative data don't mean anything. Positive data is really what we need to count on. But at the minimum, we can say that in this cohort of patients, which we're looking at with this highly, highly sensitive assay, we cannot detect XMRV, at least in the isolates which have been defined so far. Now, I want to just put in a word of caution, because we have actually gone through a real roller coaster over the last 6 months in this analysis, because when we had all these negative results, we felt, well, maybe there was enough genetic drift that, because this Tachman PCR detects single nucleotide changes, so maybe we should go do an ST-PCR, as had been described originally by Judy's group in the science paper. And we actually use the same PCR, an ST-PCR for the GAG gene, which has about the same sensitivity. If you do it in WPI cells, we can detect a single cell in negative cells. And we have gone through a very painful observation here, in that we suddenly found many, many of these viruses. Basically, every individual we looked at, we got a positive response. We sequenced these responses. We mapped them. Some were direct correlates in the mouse, some were different, some were like WPI sequences. But the final conclusion which we came to is that this is actually due to a contamination. Some reagent in our lab is contaminated, and we found that out because we then tested also samples for mitochondrial mouse, mitochondrial DNA, and also by qPCR, which is a highly, highly sensitive test. We used the second assay for IAP. This is interstitial A particles in the mouse, which is a high, high frequency. Each mouse has like hundreds of these IAPs. And with both these assays, all our human samples were contaminated. So I just want to put in one word of caution that, like, the whole story is not out there yet. The XMRV, as it's published in Science, we cannot see in our patients. We do see similar sequences, but all of these we feel like is due to a contamination. Sorry to say this. So finally, the acknowledgements. I'm at Tufts Medical School, and people in my lab have carried out most of the work. It was initiated by Albert Tai and then carried out by graduate student Brendan and with the technical help of Maddie. All our XMRV work we have done in direct consultation with John Coffin, who you probably know is one of the most prestigious person in endogenous retrovirus. He basically knows anything about endogenous retrovirus in the mouse, and his graduate student, Oya Singas, who is actually looking for the XMRV relative in the mouse. Oh, but there's something gone wrong here. So chronic fatigue cohort, sorry, I don't know what happened. It's a Mac problem. We had Susan Levine, Ronnie Taylor, and the HHV6 Foundation. Thank you.